Thanks for the opportunity to provide a brief overview of our work on whole-body quantitative imaging using the multitasking technique. First, I'd like to acknowledge Anthony for developing the multitasking framework and the technical guidance on all the applications. Multitasking is a multidimensional imaging approach that allows motion-resolved quantitative imaging without the need for ECG or breath hold. It addresses the high dimensionality challenge using a low-rank tensor model to capture the correlation between images. This reduces the degree of freedom, allowing efficient multidimensional image acquisition and reconstruction. Once multidimensional images are reconstructed, individual dynamic processes can be isolated, as shown in the middle, or quantified over all motion states, as shown on the right. Our first application was a motion-resolved T1 and T2 mapping of the heart, consisting of 6 dimensions. By replacing T2 IR preparation with spin-lock IR preparation, we were able to simultaneously obtain motion-resolved T1 and T1 row mapping of the heart. T1 row is an important biomarker for water macromolecule interactions. We can treat dynamic contrast enhancement as a time dimension to quantify first-pass myocardial perfusion. Contrast concentration is estimated by direct T1 mapping, overcoming the blood signal nonlinearity issue associated with conventional perfusion MRI. This is the work by Randy Young from Cedars. We used multitasking to resolve the dynamic process of myocardial bold signal response after the injection of stress agent regadenoson, improving the confidence of bold imaging in the heart. Another application is carotid plaque imaging by Yibin Xie of our group. Multitasking simultaneously acquires 6 types of images, such as dark blood vessel wall, bright blood MRA, T1 and T2 weighting, and T1 and T2 maps, allowing quantitative characterization of plaque features. This example shows the use of multitasking qMATCH to identify the lipid core without the need for contrast enhancement. Lipid had both short T1 and T2 values. The presence of lipid core is confirmed by conventional post-contrast imaging. This is an example delineating an area in the wall with T1, T2 values acquired using qMATCH that are consistent with the subacute intraplaque hemorrhage verified by histology. Nian Wang from our group did a cluster analysis using both T1 and T2 values, showing clear separation of tissue types such as normal wall, lipid core, loose matrix, hemorrhage, and calcification. Nian and Yibin also performed DCE imaging to assess vascularity of the carotid wall. Multitasking allows 1-second temporal dilution and 3D imaging of the entire carotid artery, which has not been possible using conventional techniques. We can then quantify kinetic parameters such as K-trans, VP, AUC in healthy volunteers and patients. Another application of multitasking framework is aortic imaging by Zhehao Hu and Zhaoyang Fan from our team. It acquires 3 types of image contrasts in one scan: bright blood, dark blood, and gray blood. This is a stroke patient with severe atherosclerosis in the descending aorta, as indicated by the arrows in the black blood images in the middle. Calcification is shown as dark areas in the gray blood images on the right, indicated by the arrows. In addition, the images are cardiac motion resolved, allowing quantification of aortic diameters and distensibility. In body applications, Zhaoyang has used multitasking to do free breathing 3D T1-T2 mapping of the liver. This is a patient showing high T1 and T2 values of the tumor. The same technique has been used for T1-T2 mapping of the pancreas, again showing a clear delineation of a tumor. Nan has used multitasking for free breathing DCE imaging of the abdomen. It allows 1-second temporal resolution and 3D coverage of the entire abdomen, including liver and pancreas. The data has 6 dimensions: 3D spatial dimensions, saturation recovery, respiration, and dynamic contrast enhancement, which can be displayed independently. T1 mapping is used to directly quantify gadolinium concentration, which can then be used to quantify kinetic parameters. Combining several maps into one produces an all-in-one quantitative map revealing the parenchyma in green, tumor in red, and benign cyst in purple. Moving up to the brain, we have now extended the technique to simultaneous quantification of 3 parameters: T1, T2, and ADC. This project is carried out by CMR in our group. ICC analysis of gray and white matters shows excellent quantitative agreement with the reference methods. This is a post-surgery GBM patient. Top and middle rows are clinical scans, and the bottom row is multitasking T1, T2, ADC maps. Green arrows point to a recurrent tumor, and blue arrows point to surrounding edema. T1, T2, and ADC maps agree very well with the clinical scans. By changing the diffusion preparation to T1 row preparation, we can quantify T1, T2, and T1 row simultaneously, and we can resolve motion. So we have 7 dimensions in this application. Here are the images acquired on an MS patient using multitasking, demonstrating the improved image quality of motion-resolved images over motion-cropped images. Zhehao and Zhaoyang applied multitasking to DCE imaging in the brain. Again, the major advantage of multitasking is better than 1-second temporal resolution, which allows accurate quantification of perfusion and permeability parameters. This is a patient with meningioma, which is clearly delineated in VP, K-trans, and VE maps. In this presentation, I have shown briefly a number of potential applications of multitasking in different parts of the body. Obviously, more patient studies are required to evaluate its clinical value. A practical limitation for large clinical studies currently is a long image recon time. Bill Chen of our group has developed a deep learning approach to dramatically reduce the recon time from 20 minutes to sub-second. We have also worked with Siemens to allow real-time preview of the images to make sure the scan is okay before offline reconstruction. In summary, due to its continuous acquisition mode and scalable multidimensional imaging capability, multitasking has the potential to be a single push-button scan for quantitative CMR. Native parametric mapping, first-pass perfusion, and post-contrast mapping can all be obtained with the same sequence framework without imaging parameter setting and without the need for ECG or breath hold. I'd like to thank the entire imaging research team at the Cedars-Sinai Medical Center for the wonderful work, Siemens for technical support, and all the grant funding. And thank you for your attention.